Perfect. Start from like all the gang. Excited. Yeah, we have to all together. We don't see them walking around anymore. Right?
We're gonna see each other. I feel like I won't do it until kind of be like, okay, you know what time it is. I do that so I do that for like a week or I was like,
Yeah, you Hi, Pink and Pinker. Saw me yesterday. I was like, we're gonna see you guys. No, Saturday. Saturday, I guess we're recording. Yeah. Friend got it for me. He's like, yeah. Here you go. Saturday? Yo, Richard was supposed to get back on us one day.
Richard.
Yo, Richard.
Please.
I thought you were gonna show us the place. Or whatever. No.
Yeah, we're coming downtown just for you.
Wait, so do you want me to just grab you later after work? Or?
Not that we might have to just do stuff.
Well, I mean, we can find out the place. It's not that hard. I mean, we're going to see some places UFC. Like, I think Knox should be open on the weekend. Wetmore, I'm not sure if it's really after it, but sometimes it's even unlocked. Or even like just outside of Wetmore. Just sort of outside. It's kind of weird. Just go table. Let's go to the upstairs. So the exam Tuesday. I mean, we're doing like go bars really early. So that shit works. It's not like I guessed it. I was
Did you get your presentation, Mark Schenker?
You can check it.
Okay, folks, if you don't mind, I'm going to try to get going. It is going to be it's going to be a little bit of a shorter lecture, so if you have to leave a little early, we're probably going to end at around 4:30-ish, I would guess, depending on how things go and what kind of questions you have. Just before I get started, I wanted to double check how you guys are doing on your grant proposals. I've only seen 2, so I'm not really sure how you guys are doing. Are you guys doing okay? Don't know yet? Not yet. Okay. There has been a motion to extend the deadline once again to Wednesday. I think would be like 5 o'clock on Wednesday the 13th, which I would be okay with if you guys are amenable to that. Okay, that cuts into your final exam study period. That's your choice. You can submit it early if you're done. As I've mentioned to you in the past, I'm happy to take a look at your grant proposals. If you share it as a Google Doc, I'm happy to take a look at it and give you some comments back on it. The emphasis is really not on trying to take away marks from you. It's really to give you tools to become really good scientists. And so I want you to think very carefully about it. If you need the extra time, certainly the Wednesday at 5 o'clock deadline is really not a big deal for us. We'll still be able to finish grading and evaluating well in advance of your final exam, which is at the very end of the exam period. Okay, is that okay for everyone? So Wednesday the 13th at 5 o'clock. I'm hoping that's enough time, hopefully. Okay. Okay, so in the last couple of lectures, I'm just going to start wrapping up the course. Actually, when I put together the final exam, there's a lot of material, and Thursday I'm going to try to go through as much as possible how you should start studying and how you should be preparing, setting aside certain office hours for you in case you need to drop in, and then taking back some ideas from you in terms of what sorts of things you want to do to get ready for the final exam. In addition, one other thing before I get going, most of you, if you haven't checked already, the marks or the evaluations from the group presentations are now posted. The TAs decided this year that they would try to give you audio feedback, and we're trying to give you the individual recordings for your group. So we'll try to send those up via email in the next couple of days, but your marks are all available for you now. If you don't see a mark, let me know. And I know that for some of you, because you missed a couple or one of the presentation dates, your mark might be a bit lower than some of your groups, and that's primarily because of attendance as part of your participation mark. Okay, so if you have any issues, again, feel free to contact me. I'll be happy to go through that with you and make sure that everything is fairly evaluated for you. So as neurobiologists, and this is really important because it is actually a really big part of your final exam, and you might think that I'm artificially trying to get you excited by RNA, but the reality is when we start looking at the genome and we start looking at different things that regulate the genome, when we look at epigenetics and we look at RNA, this is actually the most exciting part of molecular biology right now. It's not to do with DNA, and sure, lots of things are going to be found about the intron, lots of things are going to be found about histone acetylation, deacetylation, and all of that stuff that you learn in third year. That's great. But the reality is this is the future of what is going to drive neurobiology. We already know this. We already know, for example, that when cells are dying, And when cells are undergoing apoptosis, especially neurons, they release packages of RNA. And it's really weird that they release packages of RNA. And these exosomes that they release that get out into the extracellular matrix and then also affect nearby neighboring neurons is also a really hot topic right now. It's just beginning. You're seeing the creation of a brand new science related to RNA. So we're going to go back and take a look at this. And this is sort of an exciting area. And this is also an exciting area in a different way. So a lot of people have been trying to understand IQ. IQ, again, there are different metrics that you can use to assess cognitive skills, but there is no consensus. Like if you look at a genome, and everyone is describing a genome, if you get different geneticists to tell you what genes are involved in IQ, none of them can agree on this. And you have to wonder, if they're using the same sorts of techniques and they're using the same sorts of end results, why are they all coming up with different types of genes that they seem to be important? So I'm going to talk to you a little bit about RNA processing. I know many of you who've taken other courses with me previously have had a lot of the C. elegans model. I'm not going to go into it extensively other than to mention a few things about the nematode model system and why we use it in this context. And then what I wanted to go through is this idea that your body is going to be exposed to, or your neurons or your cells are going to be exposed to these exoRNAs, these exoRNAis. Why might that be really important? So a cell dies, a nearby cell dies, it releases an exosome package which contains little pieces of RNA. Why is that really important, right? Probably isn't. In the old days, we would think that's, it's just part of the degradation process that neuron is undergoing. But this exosome and this idea of exoRNAs versus endoRNAs is really important. And that's one of the main messages I want to get across today. And this paper that came out actually tries to help us understand why these RNAs and how these RNAs can get inherited and how they can actually influence future generations. And again, it's in C. elegans. I'm going to show you a little bit of a cop-out later on, but I'm going to take you through some of the evidence. And we need to understand this. We know that there are epigenetic mechanisms. All of you have studied epigenetic mechanisms like methylation of DNA, histone acetylation or deacetylation, and binding or unbinding DNA more tightly. And we know that the modifications in the genome themselves tend to be very, very dynamic. If you were to take snapshots of your DNA at any given time, it would look different. It's just like your synapses in a lot of ways. Your synapses look different from second to second. So does the epigenetic tags that exist on your DNA. So we know that these are relatively short-lived. They're very dynamic. And we need to understand if this actually plays a longer lasting signal, if there is something more than a short-lived signal that goes across. We know from some animal studies that this epigenetic tag, whatever it is, and this paper tries to address this, actually can go across generations. It's transgenerational. It's passed from the parental all the way down to maybe the 4th generation, or in some cases, the 80th generation. And this is taking place even though in theory, and I had this discussion with Dr. Eubanks just before he went to lecture last week on Thursday. I asked him, well, if these things are really being reprogrammed before the genetic message gets passed along, and all the epigenetic tags are in theory removed from the germline cells, how would you try to explain this? And again, the only answer he came up with was really it has to be at the level of RNA and proteins, which makes this a really, really cool story if this story turns out to be true. Because in a way, since we're studying neurobiology and we're studying memory, in a way it's like DNA has memory associated with events. And this programmable transgenerational memory that gets passed along is a lot like you passing along a memory verbally to your children. And so it makes it actually quite an interesting story if it turns out to be true. I sort of left this with Andrew Fire and his resilience in terms of research, not giving up, trying to figure out that this double-stranded RNA, all the experiments that he was doing weren't working. He couldn't get the gene to be expressed because of an RNA interference mechanism that no one had described prior to 1998. And this led to this idea that RNA can actually turn off genes. So if RNA can turn off genes, an RNAi is formed from double-stranded RNA, I want you to keep that in mind as we go through today's lecture. So double-stranded RNA, way back in the 1990s, was found to regulate the expression of genes, especially in a model system like C. elegans that we're going to come back to. So again, I didn't spend a lot of time on this particular diagram. I'm hoping most of you have taken either Biochem 210 or perhaps Biochem 311, and you guys have learned about RNA processing and how different forms of RNA can get processed into different species. Now, again, I'm not going to ask you to memorize this entire diagram. RNA is a very complex macromolecule, and one of the things that we're interested in terms of neurobiology, because we think that this is actually really important in terms of gene regulation, is microRNAs. And we know that microRNAs, for example, are important in Parkinson's disease, it's important in Alzheimer's disease, and they also will bind to and interact with these other proteins known as Argonaute proteins that you see here. Now, there's an important distinction. There's endogenous RNAs, double-stranded RNAs that get processed. That's normally occurring in your body. That occurs all the time through this certain pathway. This endogenous siRNA or double-stranded RNA gets processed, and we can form all of these other sort of smaller processed RNA molecules like siRNAs or hairpin RNAs, etc. And all of these interact with processing proteins like dicer, or they'll interact with stabilizing and transport RNA molecule proteins known as argonautes. Again, keep all of that in mind. Argonautes in this context are really important. They bind to RNA once it's processed, and this RNA binding can then allow it to be transported or stabilized or shuttled around to different areas. For the time being, I'm going to avoid sort of the Pee-wee interacting or pi RNAs. Again, it requires interaction with proteins, and because they're easily degradable, tend to get associated with RNAs. So we'll just keep that in the back of our mind. Okay, so we have the possibility of RNAs being developed that can be RNAi-like interfering RNAs, either from exogenous sources, like with Andrew Fire, or they can also form endogenous RNAis through an endogenous pathway that you see here. And this is the endogenous pathway. Okay, so we have two sources of potential RNAi, exogenous versus endogenous. They all use the same sort of pathways, and again, keep that in mind. Any questions on this or anything that I can clear up for you now? Because this becomes important when we start talking about today's paper. Yeah, Hiran?
Sorry, what's the exogenous pathway?
We would have to provide the RNA in whatever form. So I'm going to show you a model for C. elegans where you're giving C. elegans RNA from exogenous sources. For example, it could come from another cell. So nowadays we look at cancer and the spread of cancer where RNAs can be passed from one cell to another, and that would be an exogenous source. Cell A isn't producing the RNA inside itself, an endogenous source. It's coming from outside of cell A. And it could come from cell B, or it could come from totally outside of the body. Okay, so we know that there is heritability. There are things like chromatin modifications, and we know that these can produce heritable effects. They can actually be passed on from one generation to the other. As I mentioned before, there are associating proteins like the Argonaute proteins. And again, for today's paper, it's really not important to keep these different. I'm not going to go through all of the data in their paper, but they actually distinguish between HRDE1, which is the heritable RNA deficient 1 Argonaute protein, or the CSR1 chromosome segregation and RNAi deficient 1 Argonaute protein. Both of them are Argonaute proteins. Both of them take part in exogenous and endogenous processing, transport, and movement of RNA. And these associate in the past in other papers. If you found RNA from the parent and you found RNA again in the offspring, they were often associated with these particular Argonaut proteins. So chromatin modifications exist. Chromatin, again, requires proteins associated with it. Here are proteins that will also associate with heritable forms of RNA. Okay, so any questions about that at all? No? So we think that this mechanism might be really important. There is no firm evidence, there is no firm proof of this. It's just an observation that's been done over the past 4 or 5 years that suggests that there is a method by which this can actually occur. One of the reasons why most of these papers actually work with C. elegans is that we can actually manipulate exogenous forms of RNA really easily in C. elegans. So C. elegans, if you have an exogenous source of RNA, double-stranded RNA, and that's exogenous. It's exogenous because we can either inject it directly into the worm, we can actually get exogenous double-stranded RNA into the worm simply by feeding it double-stranded RNA, or you can actually even take a bath full of double-stranded RNA, soak it, and we'll get inside the worm as well. All of this is an exogenous source of RNA that will go inside, will get cleaved by dicer, it will then interact with those Argonaut proteins, and these in turn can actually trigger some of those heritable responses. So it's an exogenous source that gets processed like normal RNA does, and it will then be heritable and passed along to future generations. However, what most people have actually observed is that if you do this, and it works, it's been known since the early to mid 2000s, that for the most part, if you were to introduce an exogenous RNA and it was transmitted from the parent into the offspring, that this effect where it was suppressing a gene, for example, it would disappear after about the 4th generation. And there's no real explanation for this. It was one of those phenomena. It's just like saying if you come downtown, you're going to see lots of food trucks. We don't know why, but lots of food trucks are here in the downtown core, but there aren't a lot out in Scarborough, for example, or out in East York. So it was just an observation that was made. And it was also shown that in every generation, some of those worms actually lose this silencing that they saw with this double-stranded RNA. So if you injected double-stranded RNA, you could turn off some genes, but after about 4 generations, the silencing tended to disappear. And it was shown that in every generation, you would lose some of this heritable silencing. However, if you try to do some breeding of these worms, one of the things that could happen is that if you selected for ones where you saw a downregulation of these genes and you saw that it was continuously downregulated and you crossed those worms that had the exogenous RNA mediated gene silencing, this effect that normally disappears over 4 generations could actually last up to more than 80 generations. So there's obviously more than one mechanism going on. So this mechanism, if left by itself, is going to disappear in 4 generations. If you intervene and you actually select for those different C. elegans that actually maintain the loss of this gene and you select for them, something is also being transmitted along with them, and that heritable factor. You haven't knocked out this gene. You've just crossed a gene that was originally being knocked out by double-stranded RNA. Something was being carried on up to these 80 generations. No one knew what that factor was. It's just, again, sort of another observation that people had made. So 2 different things. One, normally, if left alone, that double-stranded RNA-mediated gene silencing disappears by itself. If you intervene, that double-stranded RNA-mediated gene silencing, you can actually strengthen it and make it last for longer periods of time. Are you guys all good with that? So this is the purpose of this paper. How do we find this mechanism? How do we find the mechanism that allows for this transmission to occur for long periods of time? What is it? How does it actually work? So they didn't really know what it was, but as far back as 2008, so roughly 7 to 8 years ago, there was thought to be some sort of RNA-mediated agent, whatever it was. It was a protein, it was something to do with RNA processing, whatever it was, that allowed for RNA to silence a gene, in other words, RNAi, a gene silencing through RNA, that was inherited. They didn't really know what it was, and what they thought was that this factor, every time it was transmitted to the next generation, it got more and more diluted, and eventually, whatever this RNA agent was that allowed this gene silencing to occur, or this RNA I-mediated gene silencing, it would eventually disappear. It becomes so weak or so diluted that it wouldn't be effective anymore. However, when you go back and you do the math, this passive dilution of this through transmission over several generations, it actually doesn't make a lot of sense. That C. elegans nematode, because it's a hermaphrodite, can actually produce about 250 eggs in its normal life cycle. So if it's being diluted out even amongst the 250 eggs in that first set of progeny, that means that the dilution factor wouldn't possibly be able to carry on even into the first generation. And so there was a lot of disconnect. They could see that you could strengthen this factor over 80 generations. They also know that it disappears, and it's likely being inherited, and it's likely due to RNA, but there's no idea as to what it possibly could be. It can't simply be through dilution because it would already be diluted out in the very first generation. Any questions about this before I go on? Yeah?
When you say strength, do you mean it's actually coming from the aspiration or just like
I would think in this context it means persisting, not disappearing.
and then making it stronger to the next last generation?
I don't know if you can say it would be stronger. Once you've silenced the gene and you no longer see the gene product, I mean, you can't really say that it's stronger. I think saying that it persists longer or that it's a more persistent silencing would probably be better. Okay. Okay, so again, there are lots of different things that these authors wanted to try to address. And it's great because I want you to have an idea as to the mindset. How do you actually come up with scientific questions, right? At some point, if you're going on in research or you're going into clinical research, you're going to have to come up with your own hypothesis that you can start to examine. And in order to do that, you need some background literature and you need a hypothesis. So what is this epigenetic response? Is there this RNA factor, an RNA-mediated factor that becomes less effective at a certain rate? So by the fourth generation, what is this factor that tends to disappear? Why is this RNAi-mediated inheritance that normally turns off a gene, why is it decaying instead of also being perpetuated on a longer level without you interfering in it? So one of the other things that was really good about this paper, they did a really good extensive literature research into the background, and that's something that we're all looking for in your research proposal. So with that extra time, make sure that you can, in your rationale, make sure that you've indicated that you've looked into the research, not just 5 years ago or 6 years ago, because the one thing that you can tell from all of these different types of studies is that the science that we knew 5 or 6 years ago is actually outdated, right? So we are always looking to see if your research and your background literature research is up to date as well. So there was a mechanism that suggested that if you had another sort of exogenous RNA or a secondary RNA, that this would actually lead to increased amplification of this epigenetic response. It wasn't really clear what was going on, and it might actually lead to the stable silencing or long-term silencing of certain genes by one of those Pee-wee interacting RNAs, one of those other small RNAs within C. elegans. So what is this mechanism? And this is basically the what the authors are trying to do. So this gets back into all the stuff that I talked to you about before. Right now, DNA sequencing is sort of getting faster, it's getting a lot cheaper. You should make sure you know all about the different forms of sequencing for your final exam. But the reality is more and more labs, including neuro and genetics labs, are actually turning to RNA sequencing. This is really where the future of genetics and the future of neurobiology really is. So we've talked about RNA sequencing before. Some of you have asked, do you only get mRNAs? In fact, you can isolate only for mRNAs by using poly-A probes, but you can actually get all the RNA sequences out, including those endogenous siRNAs. So before they actually do any of the wet work, they actually go in and take a look. When they go in and look at the different types of endogenous, normally occurring siRNAs, so RNA gets processed, the endogenous, normal, internal forms of RNA get processed, and they see lots of this stuff. They see lots of endogenous RNAs. Much more than they would normally expect within the germline. And if they go to ones that they know get inherited, so they can track, like in C. elegans, one of the great things is you can have all sorts of genetic information. When you go and you the endogenous siRNAs that normally get transmitted from the parental to the offspring and then into future generations, they also see that this is enriched quite a bit as well, like almost fivefold of an enrichment just of the heritable endogenous siRNAs. So it sounds like these siRNAs do get inherited, do get passed along, and they will align in the way that we know will turn off certain genes that are involved in biogenesis or normal biological activities. So this becomes their secondary question. So is this actually the mechanism? So do you inherit these siRNAs? Is this the actual mechanism that leads to this form of gene regulation in terms of RNA inheritance? And is this really what we're supposed to be looking for? So I think one of you had asked a little bit earlier on about how to actually distinguish between exogenous and endogenous RNA. So this is a normal sort of agar plate that has C. elegans on top of it. And what you see under the C. elegans is the bacterial colony. So they sit on top of this plate, and on this plate we grow bacteria, and as the worms move along the top of this plate, they will eat the bacteria as they go along. And each of these squiggles represents those about 1 millimeter in length C. elegans, or those nematodes that you've heard about previously in other lectures. Okay, so one of the things that you can do, since they're growing on bacteria and they eat bacteria, you can actually put in the bacteria different types of genes. In this case, you can put in a gene that will actually encode for a double-stranded RNA that will actually be the antisense to the normal GFP. So these worms here all have GFP. They all have GFP in the germline nuclei, and I'll show you this picture in a moment. And if it doesn't affect them in any way, it's just they have GFP, green fluorescent protein, driven by the PPI1 transgene that only allows expression in the germ cells nuclei. Okay, so you don't see it anywhere else. The PPI1 directs GFP only to the nuclei, and you'll be able to see this in this next diagram. Well, hopefully it's a little better on your handouts. So this is a typical C. elegans. These are the germline cells that you see here. This worm has been fed on control bacteria, so there is no double-stranded RNA that will knock out GFP. So this is why we see GFP in the bottom panel. On the top, though, they were fed with the anti-GFP double-stranded RNA producing bacteria. And when they eat that, the double-stranded RNA, which is exogenous, gets internalized, and then it gets all the way into the circulation of the C. elegans. And you can see that we don't see these little clusters of the germline GFP anymore, because the double-stranded RNA has wiped out the GFP expression in those cells. Is that okay for everyone? Yeah? So exogenous RNA can alter gene expression, which is what is being shown here. Nothing new. This has been done before. In fact, we do this in our 4th year lab experiments. In HMB49, we set up these lab experiments previously to be able to show students how easy it is to do these manipulations. Okay, and again, if you do nothing else, if you just do this, and you take these worms that have been fed this anti-GFP producing double-stranded RNA, which has normally turned down the expression of GFP in the germline cells, and then you wait, and you'll see this GFP expression coming back in its progeny. This RNA effect, in other words, the inherited silencing mediated by RNA, disappeared over 4 generations. Again, nothing new, confirmation of a previous hypothesis. This is their model system that they're going to be working with, and this is a really simple model to be able to use as well. Any questions about this before we go on? Okay. So this is that first RNAi trigger. They have knocked out or been fed control bacteria, either one. And this RNAi trigger normally produces a loss in the germline cells of GFP, which is what we see up here at the top. So after the parents had this, and you could see that the GFP expression disappeared, the progeny were then transferred to other plates that actually expressed either another control empty vector or that had different triggers, not GFP anymore, okay? So they have another double-stranded RNA in the other plates, but it's not against GFP. Is that okay for everyone? Yes, no? Now's the time to ask. So the parents were affected, their GFP levels are gone. Now you're taking the progeny, you're no longer treating or having bacteria that are targeting GFP, but you do have other double-stranded RNA vectors that act as secondary DS double-stranded RNA triggers, okay? We're good to go? So no GFP down here, only in the first generation. So one of the surprising things, and it was a really simple experiment, but it was a really nice experiment. One of the things that these authors were able to show is that when you look at the offspring that were hit by a secondary trigger, that one of the things that they saw was that they actually saw much stronger silencing. So this is the normal proportion of GFP expressing worms. The higher the number is, the better off, the more GFP you see. The lower it is, the more silencing of GFP that you see. And this is the parental generation, so the GFP gets knocked out. And you can see it starts coming back over subsequent generations if we don't do anything, which is what that is shown in this black bar. However, if you have a secondary trigger, it's not against GFP, just another double-stranded RNA. In fact, that actually causes a much stronger silencing of GFP in the subsequent generations. This is actually surprising, right? The effectiveness of this is not contingent on your double-stranded RNA targeting that specific gene. It's just having this double-stranded DNA trigger. And again, they did a number of other controls. No additional anti-GFP DS RNA triggers, and just the dsRNA on its own did not affect GFP levels either. So you needed that GFP in the first generation, and then you needed a secondary trigger for this sort of plot to work. But it works really, really well. They're not really sure why, because again, it doesn't make a lot of sense. You want to target GFP, so you feed it double-stranded RNA to produce RNAi, which turns off GFP. That makes sense. It's something gets inherited, which also makes sense, and it slowly washes away over 4 generations, which again makes sense. But here they're saying, if you have knocked out GFP originally, and now you're targeting another double-stranded RNA, not GFP, you still see an effect on GFP. Something else is going on in this particular model system. Okay, and again, this is just showing, as I mentioned to you before, if you look at the number of animals over time, that we will actually see fewer and fewer animals having the knockout of GFP. But if you have a secondary trigger, actually out to the 4th generation, more animals actually have the anti or the loss or knockdown of GFP. Danny.
they ever attempted to, like, if the RNAi or what they're attempting to silence have contradictory effects or opposite effects? Like I know you can't do it in this experiment, but let's say like you had one that the first generation silenced GFP, and
Yep.
then the second RNA, the secondary triggers, was actually intended on overexpressing GFP. So they have opposite effects, but it still causes secondary effect of silencing GFP, or like does it really matter what the secondary RNA is? If it contradicts the first one?
So long story short, no, it doesn't. So I'll get to their data on this. So they wanted to show, like, is it any old RNA that can work? Your question, though, is an interesting one. Not to get too sidetracked. In order for you to do the type of experiments that you're thinking of, where you're knocking something down, then you're adding a double-stranded RNA that, in theory, is a double-stranded RNA that could overexpress something, you'd actually have to encode for something that normally would be, yes, and that is not something that they have in this model system. It's an interesting experiment, and to be honest, I think it'd be a pretty cool experiment, but I can also tell you it'd be a really difficult experiment to do. So if you have about 4 or 5 years of your life, and you happen to be in a really good lab with lots of molecular cloning techniques, I would say try it. I'll help you get into that lab. Okay. And you'll come back in 4 or 5 years and tell us the experiment didn't work. That's okay too. Okay. So the simplest explanation for this is that this RNAi mediated response that's being inherited can extend this duration of this heritable RNA mediated phenomenon that they saw earlier. It's just against a different and unrelated gene. And that's their bottom line, that it doesn't matter what gene you're targeting. As long as there's something in there, as long as there's a double-stranded RNA, whatever it is can actually cause the continued silencing of that original gene that was silenced with the double-stranded RNA that you set out to mediate that RNA response. Okay, so these double-stranded RNA triggers, the second one, not against GFP, but the secondary one, just against any old RNA, they wanted to see, is it really, just as we're chatting very briefly, is it really any old RNA? So can you do ones that are normally found in the soma, somatically expressed genes like DPY2, or ones that are specifically targeting the germline genes? Remember, this is a germline gene. It is being targeted to GFP, which is only expressed in the germline by that PyP2 promoter. And so they wanted to know, is this a general effect or is it a specific effect? Is it any old RNA? They wanted to test that hypothesis, which was on that previous slide. So they looked at 11 different double-stranded RNA secondary triggers from different types of genes, somatically expressed genes or germline expressed only genes that function on a number of different cellular processes, and that by themselves should not directly influence GFP, because that's our output. We want to know, is GFP being turned off? And so when they look at all of these, for all of these secondary triggers, for all of these different double-stranded RNAs, all of these seem to be equally effective. Doesn't matter if it's somatic, germline, whatever it is, seems to be equally effective in increasing the length or the duration of that silencing of GFP that was encountered in the first generation. It didn't wash out, in other words, by the fourth generation. Is that okay for everyone? It's really kind of a neat story if it turns out to be true. Okay, so again, in all the experiments that they looked at, that somatic anti-DPY2 second dsRNA was found to be more more potent than the anti-mCherry second trigger in terms of enhancing. They were all effective, but this one seemed to be more effective than the other 10. And that there were a number of different potency effects that they also looked at as well. Okay, so Again, one of the things I like about this article is that they have this really cool toolbox that they can do a number of different things to go back and examine the GFP gene. So in theory, when you're actually causing this anti-GFP mediated response, you get all of these little double-stranded RNA mediated RNAi, which will theoretically bind and turn off the GFP gene. So you get all of these different species of RNAis, in other words. And when they go back and they take a look at this RNAi from the first generation, the one that you set out to actually inhibit GFP, and if you look at all the different RNAi species that get generated from the primary response against all of the different GFP RNAis from the subsequent generations, so the one that got knocked out originally, do you still have the same species of RNAi in your second generation, third generation, and fourth generation? And one of the things that they found was that, yes, indeed, whatever it is, with even with a secondary trigger, you're still producing the same species of RNAi that you had against GFP with this secondary unrelated trigger. So this is, again, kind of counterintuitive. Why would you actually set up to produce an anti-GFP RNA when you're actually introducing a totally foreign RNA in your secondary trigger? Again, it's one of those things that is left hanging a little bit in this particular paper. But one of the things that they're suggesting is that the reason why we have this, the reason why this mechanism actually exists, is that if you turn this RNAi mediated response to turn off a gene, in this case it's GFP, it's doing so because we've turned on the exogenous siRNA pathway that I started out today's talk with. We have an exo and an endo. They work together, they work in parallel. And so this RNA mediated response that turns off a gene through this exo siRNA mediated pathway is really important. And what happens is, by enhancing the production, because you're introducing it, because you are providing these exo siRNAs, what ends up happening is that somehow the normal endogenous small RNAs that normally get bound to either CSR1, one of those Argonaute proteins, or HRD-E1, another Argonaute protein, that normally bind to other siRNAs, you're now disrupting this regular pathway because you're providing an overwhelming amount of this exo siRNA. Now you have this difference. You're changing the balance with which these RNA populations can exist. So you've turned on the exo siRNA pathway, and a feedback response actually will return this back to its normal levels. Over time, there's a feedback mechanism to turn this exo siRNA pathway off. However, this off state that normally would occur, and they don't have a really good explanation for how it goes back into the off state, you can prevent it going back to its default off state by actually having a reset by introducing another double-stranded RNA. It doesn't matter what it is. It's just another double-stranded RNA. As long as that original pathway that you started off with here, as long as this exo siRNA pathway is still turned on and it hasn't completely disappeared, you can go and prevent it from being turned off by introducing this transgenerational timer by any old double-stranded RNA. The mechanisms aren't all clear right now. They don't really know, other than it's likely involving the exo siRNA system. It's likely because of this pathway not being reset. But how all of these different molecules work, and again, you don't have to recapitulate or worry about the diagram on the right. It's more important to concentrate on the bullet points on the left. That they're not really sure how this mechanism works yet. It's a really cool phenomenological description, and it's part of a model, but they really don't have a lot of a finalized picture yet. It's more of a, we're about 80% of the way there. This is what we've gotten so far. It's a really cool story, but there are some future directions that we obviously have to take type of story. And I think that one of the reasons why they're doing this is they're literally trying to get that first publication out. Danny?
Does the secondary trigger still function as a primary trigger for whatever it's doing?
You can also show, and they show, not me, but they can also show that it also acts as a trigger as well. Yes. Mhm. To be honest, I took this from their paper, and I actually think that it's one of those things that I hate seeing. I really do. When you ask students, when you go into your fourth year classes and you ask students, so what's the next experiment that you would do? So they did all of their experiments in C. elegans, right? So the next experiment would be to do this in a higher organism, like a rat or a mouse. I hate that. Like, that's the worst future direction you can ever give, right? Because that's like a default um, thing. Like if you're watching a lot of TV and you really like watching a certain type of sporting event, and you're watching the NCAAs, of course you're likely going to watch the NBA finals, right? It's a lot like saying that you've already done this, you're just going to do it on a bigger scale. And I'm not really sure that that's a logical expansion here. And again, it sounds really good, and the way they write it is really really good. Looking at the epigenetic mechanisms, if there are any epigenetic mechanism like chromatin modification, which they didn't look at all. If any of this can actually influence this RNA-mediated pathway in higher organisms. Again, I don't think it's a really great future experiment. Again, it's a really cool idea because in theory, it might mean that it's actually not a good thing to have. It can actually be, in theory, deleterious to maintain these epigenetic responses that go from generation to generation. So something killed off your parents, if they left an epigenetic tag, that thing is no longer in the environment around you, and actually might lead you in this danger-free sort of time to be more at risk. And so it's possible that since this is a generational effect that you're seeing, that it actually might work against you in the long run. And again, whether or not this is adaptive in higher organisms, again, I think it's a little bit of a cop-out, to be quite honest. I don't think that this is a really good sort of future directions to be implemented like in a Cell paper. Danny?
Constantly bring a secondary trigger to, I guess, reignite the first silencing effect. How does that ever, like, how does that first silencing ever go back to a whole, like, how does it ever not get silenced again? Because in the future, I'm sure that more triggers will come in, and constant second and tertiary triggers or quaternary triggers. Doesn't it constantly keep that primary trigger always silencing? So won't genes, like, throughout time just become more and more silenced? How does it ever go back to not being silenced? You can't really, like, you just feel like the effect compounds without ever seeing it ever go back to a balance of not being silenced.
Not sure, Danny, to be honest. I think that if you were to have the organism adapt through some of these RNAi-mediated gene silencing events over generations. So again, they have a really short generational time. So they live about 3 to 4 days. So they might be able to adapt, for example, relatively quickly to a lack of resources like food in their plate. But I'm not sure, like over the long term, whether or not that would lead to a permanent silencing or if it's a permanent effect or not. It's an interesting question, to be honest, but I don't know.
Like when we think about, well, we have epigenetics happening in us the same way, and
Yep.
throughout our history, if anything, again, I know we can't just absorb the RNA, but if anything could just make you silence something that was silenced.
Yeah, I think those are really good questions, but let me just take a step back. These epigenetic factors are different. We're not talking about the type of epigenetics that maybe more of you are familiar with, like a maternal rat starts licking its offspring and causing these epigenetic tags to occur on the offspring. So that's like one form of epigenetics. Here, what we're talking about is if, for example, you're exposed to a virus, or you're exposed to a cell dying that contains like a group of RNA, that's going to affect some of your cells. And that might last for you know, several different generations. So we don't really know what the overall effects are. These are exo RNA mediated effects. We don't really encounter, as far as we know, we don't know everything yet. As far as we know, we don't encounter them except in cases like Cancer, we encounter these. In theory, when your brain undergoes neurodegeneration and some of them start dying, they release exosomes. These exosomes contain RNA. So we always talk about these pathogenic mechanisms, but in theory, like what might happen is some of those neurons might be affected in the long term. So you ask these types of questions that what might be the long-term effects? So again, if I'm just being very, very speculative, right? In theory, let's say someone had the beginnings of a neurodegenerative condition, and some of these neurons started dying, and some of these exoRNAs, the double-stranded RNAs, were then passed along. The responses and the genes that got turned off were passed along to their offspring. And they might also have inherited that. They might encounter similar situations. And so you kind of wonder, how do these spontaneous mutations that lead to things like prions, sometimes where we can't find a genetic alteration, how do these types of things occur? So as I mentioned in other neurodegenerative lectures, most of the neurodegeneration is sporadic. We don't really know the cause. But let's say there is a cause. Maybe it is mediated through longer term epigenetic factors, right? We don't know. So it's like a brand new field, as I said. Like you can literally make your own name in this field if you have a good idea about what these could be used for. Okay, I think actually now is a really good time to take a break. We'll go through this paper. And this paper is actually really interesting in the sense that it's less of a genetics and less of a neuro paper than it is a statistics paper, which is what most of genetics is kind of moving into. So let's take a break and then we'll come back and spend about 20 minutes going over this particular lecture.
Hi. Okay, so
So why do they have this? You said that they actually upregulate the silence. Yeah. Well, I have to. I would expect the M-chair to silence as well, like the silence.
My understanding is that there's probably a common, like it's like you said, the sweet spot. Yeah. Probably because what seems to be going on is that there's a feed forward mechanism. It seems like the point of the trigger is to feed forward and upregulate pathway that's involved in the process. So the triggers are always going to be obviously there are more things that you can do. Which is why we should test this in higher orders. Such a problem. Okay, so I'm going to work on I'm working on it today. And I have a fair bit of so I have a few ideas. I want to scrap the future graphics. I want to show because I'm really excited about that. Yes. She should be and also, this is like, right now, this is just something God help now. You know, the process, instead of looking at yeah, that makes everything yeah, you can use that for you can stage I know. That's a good question. And the answer is not very clear. But I think exactly, because you're putting it in well, I know that weird. of stating that they already want like I said, I don't think it's necessarily sleep. The other thing, for that, I was thinking we should just use the I don't know. I would look on that's why even if it's just excavation, we can still look at excavation. That's not necessarily a problem. You still have I wouldn't think. Oh, that you keep it in there, and actually just have I don't want to do it. Just I think it's more important to confirm that this is there. If we were going to do that, I assume we want to state this to measure.
Okay, so there are lots of interesting questions, discussions about how to write a grant and what sorts of experiments that you should do. Again, I'm hoping that with that extra couple of days that you will come up with some really good hypotheses. It has to be hypothesis driven, has to be biology based, and it has to be something that you can actually demonstrate that you can answer that question or that hypothesis. If you have a hypothesis that you don't have a good way to be able to answer, it's not a really good grant. And it's something that I'll help you work on because I want you to think about it and go through the thought process. So going back to thinking and about thought processes, what a great segue that is. Do you think there is a genetic basis for IQ? And if so, can you give me one example or one clear, indisputable example that intelligence is in fact due to genetic causes? there's a really simple way to answer this question. Is intelligence indeed genetic? Yes?
Well, there are some diseases that are not associated with intelligence that are genetic.
Some diseases associated with,
Intelligence.
okay. Can you give me an example? So I think you're on the right track.
Well, Alzheimer's disease.
So Alzheimer's disease, again, I'm not so sure that would be a great example of this, but you're on the right track. Sure. Sorry?
Like, like Down syndrome or
Okay. But in that case, Arslan, not to make this sort of a Socratic teaching method, but in that case, you might argue that because you're fundamentally changing the structure of the brain, and the brain itself is malformed, then that might intrinsically cause a lack of intelligence. And maybe it's actually true, but let's say you have someone that by all appearances doesn't have Down syndrome. You know, are there diseases that actually look like that, or disorders that look like that? Yeah. So okay, Asperger's possible, sure. Arjun?
I don't know if this is answering the question explicitly, but the main ones I can think of are diseases that affect the intelligence that are explicitly genetic, ones that would like completely change the brain. Because I mean, because of the whole different definition.
Just hold on to that thought for 1 second, though. It doesn't necessarily have to completely change the brain. Okay, so let's get back to that in a second. Yeah. Rett syndrome, sure. Again, you might argue that there are all sorts of changes. There might be some developmental delays, etc. But if you were looking, and again, this is one of the reasons why I brought up this particular paper, sort of just to wrap up the course. You know, as students who are interested in both genetics as well as in neuroscience, one of the things that we think as neurobiologists and as geneticists, that if we understand everything about the genome, which is what this whole course has been about, then in theory, you would understand everything about the human body. If we understand everything about neurobiology, in theory, you would be able to describe and understand every last behavior. Because isn't that really the goal? If you can actually isolate every last synapse and how they join together, you'll be able to understand how people learn, how emotions come about. If you look at the connectivity and you understand the structures, that's really the end goal of both neuro as well as genetics, to understand everything about the human body as well as the brain. Richard, did you have a...
I was just wondering, like I was gonna suggest that maybe individuals with learning disabilities can be considered because like the brain will appear to look normal, but then obviously they have issues with understanding concepts.
Okay, so that's an interesting approach as well. So, you know, learning disability, intelligence may be moderately affected, etc. Just because you can't read well or you have dyslexia, a possibility for sure.
Rather than go through disease,
Yeah.
can't we just do based on the lines of like GWAS studies and people's and just take like
So
standardized form of-
Danny, I like the way you think, but let me ask you a question. Do you believe every last GWAS study that comes out? And does every GWAS study actually agree with each other? Because this one, for sure, this is from 2014. There are other ones that came out in 2015. There are ones all the way back in 2010. None of them agree to the things that relate to intelligence, okay? But it's an interesting idea. So how much faith do you put into GWAS studies?
I get what you're saying, that a lot of it is very, there's not a lot of, I guess, agreement on certain, the significance of certain variants.
Maybe, but this is actually the question. If you were to tell me if there's a GWAS study that everyone agrees on, okay, so APOE4, Alzheimer's, and this actually comes up in these types of studies, is there anything that everyone agrees on? Intellectual disability, actually the GWAS studies are mainly in agreement. So we know that for individuals that have intellectual disability, where we know that their IQ is affected, there are clear markers that affect intelligence there. This is going the other way. How do you show that certain genes are affecting higher intelligence? And is higher intelligence actually something that is genetically encoded? Or is it actually a combination of different factors, which again, there is no firm agreement on as well. So I'm going to take you through this paper, and we're going to try to understand this paper. You don't have to worry about all the details as we go along. I'll try to point out the ones that I think are a little bit more important for you. So a lot of different studies have used those trio studies, twin studies, familial studies. So at least some of that variation in cognitive performance, how well you do in school, at least some of that is genetic. What you get from your mom and dad, a proportion of that is genetic. People disagree, like is it like 100%? Is it 80%? If your mom and dad are really, really smart, like are you more likely to be smart as well? But there is some component to that. There is a genetic basis for how well you do cognitively. Okay, it's not 100%, we know that much. Otherwise, every last one of Einstein's kids would have been as smart as he was, right? However, we do know that it's not all genetic, but we can't really agree on all the different factors that go into this, whether or not it's a genetic basis. So a lot of people over a number of years, once we started getting better at genotyping, better at doing genome sequencing, have been trying to identify whatever common genetic variants lead to better cognitive performance. What makes you smarter? What makes someone who goes to U of T have different genetic sets than someone who goes to Ryerson or to York or to Brock or whatever it is? So how do we actually know all of these different things? With the exception of ApoE, which came out as something that occurred in older individuals that were undergoing cognitive tests, nothing has really come out, right? ApoE was actually discovered not by people looking for Alzheimer's variants, but they were looking for cognitive improvement. And when they started testing individuals that were 65 years and over, this ApoE came out as a marker for cognitive performance. It just so happened that they would also develop Alzheimer's disease a few years down the road. So a lot of people have been looking, and a lot of people have been trying to use the various approaches. In the old days, we would have used this traditional approach. We would find this one candidate gene. We think that maybe the AMPA receptor is really important for learning and memory, cognition, etc. Whatever it is, we would do this candidate gene. We would look to see, are there any genetic variants associated with the AMPA receptor that leads to better performance? So if they have this variant in AMPA, do they do better at school? Or do they do better on cognitive tests or cognitive function tests? And again, a lot of this comes from, well, it makes sense. Like, you know, the AMPA receptor, it's a known gene that is associated with learning and memory. And so these hypotheses tend to be a little bit biased. You try to look for candidate genes that are involved in things that you suspect would normally be involved in this. And in this course, we've already seen several times, for example, when we looked earlier on in schizophrenia, the best marker for schizophrenia is not, in fact, dopamine. In fact, it has to do with the immune system. And it's because people were really narrow-sighted, and they were only looking for candidate genes that had a known biological function that made sense to them as biologists. So a lot of different candidate gene associations have actually been reported with cognitive performance in small studies. When you go and take a look at like this class, for example, and you look at this class and you test to see what sort of genetic variations you have, you get a lot of different results out. But when you start looking at a larger scale, if you start taking and extrapolating the results from this class on a small number of individuals that you've identified candidate genes, and then you look at a GWAS study, it doesn't work. So again, doing those traditional approaches, a number of different genes that sort of make sense have come out. Things like BDNF, right? It makes sense that BDNF, and we've talked about this valine methionine previously, this sort of SNP, it makes sense that BDNF comes out. But in terms of intelligence and IQ, actually BDNF doesn't seem to be a big player on the wide scale. Other things related to like ADRB2, another receptor system, all of these things have been implicated on small scale studies. But when you start testing for these using GWAS, none of these have that level of statistical significance. So then how about go the other way? The ones that we've been looking at in this course. We've been looking at GWAS, right? We've been looking at, let's take two really large populations, one that is a control, one that we're looking at, one that has a higher intelligence, one that has a lower one. Let's see if there are any differences in the different SNPs that we can identify. So another research strategy is to use this GWAS that we've looked at. So we look at large numbers of individuals. If there's anything statistically really important there, across these large individuals, even though everyone has a large number of SNPs, if it really follows with intelligence and is a really big effect, we should start to see this at the level of a GWAS, at that threshold value of 10 to the minus 8th. And again, we've talked about this previously. And again, for physical and medical phenotypes, if you're looking at a genetic condition that causes a medical condition, then it works great. GWAS is great at predicting whether or not, if you have a particular SNP, you're going to die by the age of 25 the next time you go for a jog. You have long QT syndrome. You have the SNP for that. It's great for identifying things related to medical phenotype. It's great for identifying things related to height. So if you look for SNPs that are more associated with height, these types of physical characteristics, GWAS is great. But for these types of things that we're interested in neurogenomics, things that are a little bit more subtle. So for example, IQ, you might not think it's subtle, but it's actually a subtle test. It's a behavioral test. It is much more difficult to actually go back and replicate and show that this has a functional consequence. However, there have been there have been papers, this is one, that have come out looking at GWAS studies. There have been no consistent findings in the literature that say that if you have this SNP, if you have this particular variant, that you are going to have an increased likelihood of higher intelligence. Yeah, Danny.
Do you think that the lack of consistency amongst GWAS studies for intelligence comes from the lack of an actual accurate way of measuring intelligence?
That might be one. That might be one issue.
We don't know
Absolutely.
what to look for? Because, like, in the sense that we're grouping people that say these people have an IQ from a standardized IQ test greater than 145. These guys are considered smart. Now let's go see what their brain looks like. But there's so much to intelligence that it's hard for us to even measure it.
So I don't disagree with you. I think that in many ways, some of these studies are exactly like that, where if you're looking at the differences in your genome that lead you to be smarter than someone else who doesn't have those same differences, that one of the ways that we test for intelligence might be inaccurate, or it might be lacking, or the tests themselves might not really be a good reflection of intelligence. Absolutely. So you can imagine, for example, people try to apply the same thing to things like religiosity or spirituality. How do you define religiosity or spirituality? It becomes really challenging, right? So I don't disagree with you that the way that we can identify whether or not you're tall, that's easy to measure. Whether or not you are intelligent is harder to measure. You're right. So again, no major genome-wide association studies that have actually led to replicable results that led to anything that is of any use. So this paper came out in 2014, and what it tried to do, if you read through it, and it's a rather short paper, so it's actually kind of interesting to read. If you're really good at statistics, this is the paper for you. So what they wanted to do was actually look at a two-stage approach. So they say all the other studies that have gone on beforehand actually haven't worked well. And so one of the things we want to do is take this two-stage research strategy, and they call this the proxy phenotype measure. So this gets back to your question, Danny. How do you normalize things? How do you make sure that we're doing things the right way? So they do a GWAS first on a proxy phenotype. And it's done on a relatively small set of single nucleotide polymorphisms that are associated with a certain phenotype related to intelligence or related to a cognitive test that everyone takes. In the second stage of this two-stage strategy, they take these in these particular SNPs that were these candidates that they were using to help identify the original group of SNPs as a sort of stage to look at independent samples on a larger level. Now, this is where I get a little bit lost because they do a lot of statistical manipulation in this particular study, and I'll go through some of the statistics with you. So they set certain thresholds. They explain why they set certain thresholds, but there's a lot of statistical sort of hand waving as to why they are looking at things in a certain way. So again, to get back to your question earlier, Danny, so one of the things that they are very explicit about is how they are measuring cognitive performance. Better cognitive performers have a higher G value, which is a Spearman's measure of cognitive ability, and that's a standardized psychology test that people undergo, and that's sort of the test that they're using in their paper. So in order for you to verify their results, you would have to go back and do this. If you're doing another GWAS study, you would have to use their test. Even if you didn't get the same results as they did, they would argue that you didn't administer the test the way that they did. So again, there's a lot of things to keep in mind as well. So again, you don't have to worry about the statistics. They do a lot of sort of weeding out ahead of time. Before they even get to that proxy phenotype, they do a lot of these ex-ante power calculations. They want to know, based on previous studies that are in the literature, if we are looking at X number of SNPs, how many of these individuals should we have for this proxy phenotype, and then what should be the threshold that is set. And again, I don't understand all the statistics, to be quite honest, but this is sort of the ex-ante power calculation that was used to help set this particular threshold. And again, by doing so, they wanted to maximize the total number of positives that they saw, and the total number of positive SNPs that were associated with doing better on a Spearman's cognitive performance assay was about 927. So there were a lot of SNPs, in other words, that they were looking at. So again, they don't go into a lot of details, and I didn't want to read through all of their supplementary methods, but they used a number of other pruning methods, and that's sort of what they call all of these things together, both statistics as well as some just general looking at the SNPs themselves and seeing if they make sense or not. These pruning methods resulted in about 69 of these independent SNPs out of those 927 that they wanted to examine further. And out of all of those, and these are all of those 69 SNPs that were identified in that pruning method, out of all of these, one of the things that they wanted to do is to see if anything in particular stood out. Are there any of these SNPs that actually allowed those individuals that took this cognitive test to actually perform a little bit better? And out of all of those studies, they came up with these 3 SNPs. And they tried to, again, normalize for the amount of schooling. They tried to normalize for a variety of other factors. These are the 3 SNPs that they identified as being the most highly correlational in terms of IQ on that Spearman cognitive performance test. So those top 3 education-associated SNPs, the ones that you see up here, and they throw out some of these data points. So if you're wondering why this data point wasn't taken, for example, some of those were thrown out even in this analysis. When they go back and look at this 3 education-associated SNP table that you see here, they were, after this Bonferroni correction and after going through and looking at some of the different SNPs that are associated with some of the different gene loci, these are the 3 SNPs that they were left with. And they say that they have a clear separation from the others. I would actually argue that they probably, this amount of separation after all the corrections that they've done, I would argue that it's probably not valid. The other thing I don't like about this particular test, and if you're taking other genetics courses, they should actually go back and take a look at some of these other outliers, the ones that they're casually throwing away that are also equally important. If you're doing really well up at the top here, why not take a look at some of these other SNPs that are associated with not doing so well? And again, those are ones that they don't address at all in this study. And I understand the rationale for this, but I would argue that this clear separation that they're talking about, and yes, it's a pretty sizable gap, but by eliminating things like this SNP out here, they're actually making it harder to believe that their data is actually all that meaningful. So these 3 clearly separated SNPs that they've already corrected multiple times, and then they also go back and do another Bonferroni correction on this, and we talked about Bonferroni correction earlier. These are the 3 SNPs that you are left with. I would argue that if you are looking at this study and you're trying to replicate this study, based on the way that they've set up both the cognitive performance tests, the way that they're looking at these particular SNPs, this would be a really hard study to replicate. My guess is that if someone's following up on this, they're not going to get the same 3 SNPs that they got here if they use similar populations. So again, how big is this effect? So they see these 3 SNPs, they go back and they take a look at how important are these 3 SNPs. If you take a look at them all together, if you look at the R² value, and again, if R² seems like a foreign concept for you right now, you're in a little bit of trouble. You have lots of time. You have a whole month to figure out R². It's actually something that we talked about when we talked about GWAS. You need to know R². It's on your final exam, and it's something that if you're leaving a genetics course, you should make sure that you know. So this is actually the R² value is not all that statistically significant. So they just told you that they have clear separation. They're quite distinct from all the other SNPs. But the R² value from all the other published literature actually isn't that much better, right? So I told you that there are other publications that have come out, other GWAS studies have identified other SNPs, and when they report their R² values, it's not any different. It's not like their study had these great results that had like these SNPs that were far better than any of the other studies that came out. Their R² values are no different than previous studies that looked at cognitive performance. So just in comparison, so if you're looking at complex physical traits like height, where there is clearly a genetic association, and you have SNPs that are associated with height, and you go back and you look at their R² value, it's much, much higher. It's an order of magnitude higher plus some. If you're looking at genes and SNPs associated with body mass index, again, a physical trait that you can measure, like looking at height versus waist circumference, then you can see that the SNPs that are associated with body mass index are much, much higher as well. So in terms of cognitive performance or intelligence, the SNPs that we have here are very, very weak. It's not a big effect, okay? So even though they make all of these great claims about their statistics being clearly separated and delineated after all that manipulation, at the end of the day, their data is no stronger or any better than any of the other GWAS studies that have come out. And so even though they have candidate genes, the most promising genes that are near those SNPs in the loci that encode things, according, again, to this reactome, which is one way to put in all these genes and then look to see what these different genes do. Most of these different genes are involved in activating the NMDA receptor. And hopefully, if you recall, the NMDA receptor is one of those really important receptors that are involved in things like synaptic plasticity. So does it help cognitive performance? Those SNPs that are around things like Norexin-1 that regulates clustering of the pre- and postsynaptic complex, maybe it is. But I would argue that lots of things are going to cluster around NRX-N1. So it's not really clear, to be honest, if this is actually a real effect. So it sounds really great because remember, we take 2 different approaches. We have a candidate gene, then we see if it actually matches on a GWAS, or we can do a GWAS and see if we can pull out candidate genes. And this is kind of a fusion of those. They did a GWAS, they pulled out some candidate genes, and these candidate genes that they have, the SNPs, are clustered around regions that are implicated in learning and memory. But again, I would say, and I would argue that it's not a great study. So we're not any better off. That yes, we have some SNPs that are around regions that implicate synaptic plasticity, but I don't think that they're any different than any of the other GWAS studies that have come out. So other studies that have come out have actually shown that if you take a look at things like intelligence, that a lot of intelligence actually does tend to be genetically inherited. And again, that part is, I think, not under dispute. That if you take a look at GWAS studies, that there is a linkage between genetics, inheritability, and overall intelligence performance. It's not the only thing. So the red bar tells you that it's related to additive genetic effects, things that you get from your mom and dad, is in red for intelligence. But then there are other factors that also contribute about 40% of your native intelligence. Things like your shared environment, non-shared environment, where you're at school, where you're at home, your home life, your school life, etc., also play a really big role. And I think all of us can appreciate that intelligence probably is shaped by many, many different things at many, many different times, right? You might be the smartest person in the world. If you have no access to books at an early age, you're probably not going to be the smartest person in the room when you're 20 years old, okay? So those are some of the different factors. You could have started out with all of the genetic intelligence that you possibly could have, but if you don't have the right shared environment or a non-shared environment around you, no one's teaching you, you don't have books to read, no one's teaching you how to read, all those things will simply be lost, and your intelligence might still be there, but you're not going to be able to reach that full intelligence. So I think the bottom line is, when you're looking at these types of genetic tests and you're looking at really important behavioral paradigms, so we're looking at neuro and genomics together, I think there are lots of caveats to this process. And I want you to be aware of those caveats, whether it's statistical manipulation of things, and it's not invalid, by the way, everyone does it. But to me, as a traditional biologist, when people start like crunching numbers, you ever do those experiments? Some of you do research, right? You ever do those experiments where you have you've gotten results, you have like an N of 20, and then when you compare your control versus your experimental, your p-value is 0.051. You ever have that happen? And then you kind of wonder, I wonder if I can throw out some of this data, or I wonder if I should just increase my N value or increase my experimental, keep my controls the same. And in many ways, when they do all of these things, to me, it's a lot like the manipulations that you do that, again, as a pure biologist, I would argue aren't so good, right? So if you're trying to change your p-value to make it statistically significant by changing the experimental conditions, etc., I'm not so sure that's a great thing. Okay, so unfortunately, I wrote this beforehand. So now it's going to be due at 5 p.m. on April the 13th. And I'm hoping that if you can't arrange for a hard copy to be dropped in the office, you can let me know. I know that you guys are going to be in exam season. So let me know about this. There will be a Dropbox ready. You won't be considered late if you submit it by 5 p.m. on April the 13th. There will be a review session this Thursday. I'm probably going to stream if we can get it to work consistently. Yeah, Eddie?
Where would you submit actual paper?
Just in the HMB office. The HMB office, by the way, closes at 5 o'clock. So make sure that you're there by 5 o'clock. Okay, so there will be a review session on Thursday. If there's anything that you specifically want me to go over, so I'm going to give you a detailed study review. I'm going to tell you like what you should study, which sections are going to be more heavily emphasized. I'm also going to have extra office hours, and the remaining 4% of your participation is going to be as follows. It's just going to be for completion. I'm not going to grade it. I want you to see each other's work. Some of you have done this with me previously. I'm going to assign you a short answer question. So everyone in this, there'll be 5 short answer questions. You will be assigned one of those 5 short answer questions. You will submit the short answer question to me, and then I will post it anonymously. So I might receive a short answer question number 3 from Anne. I will take her name off it. I'll post it on the discussion board. And then for the other 2%, what you're going to do is just complete a response and say, Anne, that's the best answer I've ever seen in my entire life. Don't change a single thing. I'm sure if that question gets asked on the final exam, you'll get 5 out of 5. Probably not, but I'm just saying. And so this is the assigned, not saying that Anne wouldn't get 5 out of 5. I'm just saying that that's not the response you would want to give. Too late. Yeah, you're right. Okay. So the 2% completion of the short answer, and then the completion of the assigned response. Just for completion, so today's the day where we're going to decide what day do you want me to post the questions and then assign you the things to have it back by. So since you have something due on April the 13th, when do you actually want to submit your short answer questions? The 15th, the 16th? No, 25th isn't going to work. 26 is not going to work.
When do you want us for you to post?
So I'm likely going to post these questions by early next week. Okay, so when you guys have that study break on April 11th, they'll be waiting for you. All of you will have a number assigned to you. So Lauren, you might be number 1. Jenna, you might be number 2.
We email the responses to you?
They get put into a Dropbox.
Okay.
Yep. And then I just anonymize it, and then I collect all of response number 2, and I put them on discussion board. So what date? The 15th? 15th is okay? But you actually have to try. Like the 2%, it's not like, I'm lost, sorry, tried my best, can't do it. That doesn't work for me. Like you actually have to try it, right? You can look at your notes. That's one of the reasons why I want you to do it. I want you to look at your notes. I don't mind if you look at your notes and try writing the response. Richard? So this is going to be when you have to submit it back to me by April the 15th, for example. So you'll get it on April the 11th. All of you on Thursday will be assigned a number. Someone will pick one number, and everyone after that person, like for example, if it's you, Richard, if you pick number 1, the next person after you will be 2, 3, 4, etc., and just keeps repeating. And then the other 2%, as long as it's done before the final exam, then I'll be happy with that. So the 15th is okay for everyone? Yes, when it's due, yes. No, not the 21st. Okay, can you give me a date that you would find reasonable? How's that? 21st? 18. 18?
How long is the response supposed to be?
It's about a paragraph.
Okay, so like 2 days after the 15th is fine. It's a paragraph. How long is it going to take?
Okay. How about we make it somewhat, is the 18th okay for everyone? 18th? Okay. So what I will do is I will post this again on your announcements page. So your completion of that short answer question will be due on the 18th by 12 noon. And then you will have until the day of the actual midterm, final exam to submit your assigned response on discussion board directly, okay? All right, so if I can ask you for one other favor. So normally I don't ask this, but this is the first year where we have done a reboot of this course. And so normally I don't ask students to do the course evaluations. We have a few minutes now, and if you can take the time before you leave to fill in your course evaluation, I'm going to leave, so you don't have to worry about that. I don't see them. By the way, no one sees them until after the marks are submitted for a course. So you don't have to worry that, what if I give him a bad review, is he going to give me a bad mark? Well, first, it's anonymized, and second, we don't see them until the marks are actually submitted. The other thing to keep in mind is our chair also sees all of your responses as well. So they decide, like, you know, this course doesn't seem like it's working out, or maybe we need to change things around. And we want that feedback, to be quite honest. And you know, this is some of the feedback that we got from Dimitar's class last year, which led to some changes this year. And we want to know, like, should we change things up some more? If you could spend a few minutes, and I know your time is valuable, but we've ended a little bit early. If you could spend a few minutes just filling that out. Just before class, I took a look, and the response rate was at about 20%. I'm not expecting 100%. That would be wonderful. I would win the Faculty of Arts and Science Response Rate Prize. Woo! Okay, cool. There isn't, I'm just saying. Yeah. But I would love it if at least 67%, 2/3 of the class actually responded. And it really is. I swear it's really important, and it really is something that we read. It is something that does affect this course in future iterations of the course. If you think it doesn't, you can talk to Dimitar, because it does. And please provide that feedback. It is something that we take a look at. And because this is the first sort of reboot of the course, I would love to get that feedback from all of you. And I'm going to leave now. If you can spend a few minutes while you still have some time here, if you have a laptop. If you don't and you happen to do it when you go home, that would also be great. And I'll report back to you on Thursday, like what percentage of you have actually filled this in. Again, thanks a lot, and I'll see you all on Thursday. And we are going to take a picture, rain or shine, so be prepared.